Good afternoon, everyone. As mentioned, my name's Edwin Stone. I'm Head of Cell and Gene Therapy at the Technology Partnership. The organisers did rather well, I think, in putting this talk after the previous one. In 2014, I was fortunate enough, whilst at TTP, to get to work on what was to later become the first EMA-approved cell therapy. And back then, it really struck me how the biology was incredible, absolutely revolutionary, and we've really seen, I think, over the years, the maturity and the growth of that, and again, as we've just heard in the last talk, some incredible new things still coming along. But it struck me how the engineering, and I mean that in the broadest sense, supporting other technologies, whether that's software, whether it's actually human factors, how that combination of things becomes critical in achieving what we all know is vital in this space, and that is patient access. And that gap was quite worrying, and one that I was passionate, and TTP were passionate about trying to help address. Just a little background on TTP. So we're based in Cambridge in the UK. There's another Cambridge, I believe, somewhere else, but that one's the original one. We're about 280 people, and a very mixed group of scientists and engineers, those disciplines that can attack multidisciplinary projects. And we operate quite large scale. We do around 200 projects every year, tackling hard projects and large projects, classically. We operate globally. A lot of our projects are in the US, but also across Europe and the rest of the world. And perhaps one of the most important things is we're employee-owned. So we take a very long view in the things we do, and I think in this space that's absolutely critical. We're not thinking about the next 3 months or 6 months, we're thinking about the next 3 years or 6 years or 10 years or 20 years, and we invest behind skills and facilities to support that. And this is a system quite a lot of people might be familiar with. If you're not, this is the Amgen EPO manufacturing line. It was built around 20 years ago by TTP. And the rationale behind it was one of timescales. Roller bottles are often used in development, and Amgen had validated that manufacturing approach. They didn't at that point then want to stop and wait to revalidate that on stirred tank systems. And so, despite being an unusual route, the aspiration was to move to 40,000 roller bottles. And so whilst in things like autologous therapies, we talk a lot about scale out, we talk about that being a new thing. In fact, we have done it before, and we've lived some of the challenges that come with that. There, I've made the comment, 1 isn't the same as 10,000. In fact, you could perhaps even say 1 is not the same as 1 million in this case. There was over 1 million individual components in this system, all of which have their own challenges. And as mentioned previously, how do you scale? How do you move from that first proof of concept of a biological technology to achieving scale? To do that, you've really got to get on top of the things that come as you move from 1 to 10,000 to 40,000 to 100,000, the reliability challenges that emerge with that. That one prototype system that we all at some point have to have working on the desk to show the core of the technology works. We have to know what happens when we've got 100 of those and when we've got 1,000 of those. And I guess if the simplest lesson that can emerge from that is it will find every weak point. It will stress test your technology to the limit, and it will find every weak spot within that. And so it's critical that you understand that early if you're looking to truly get to scale. And to bring things much more recently up to date, we're fortunate to have had the opportunity to work with many innovative companies. Over the last 3 years alone, over 30 projects in the cell therapy space, and across a very broad range of applications, right from arm to arm, as is often referred to in autologous therapies. Just to quickly run through some of these projects that we've been undertaking. So Achilles Therapeutics might be familiar to a number of people in the room. That's a TIL-based therapy. For many of their processes, existing equipment does exist. It might not be perfect, it might not be what you might choose in 5 or 10 years' time, but it will be sufficient. But for certain parts, things do not exist today to help them undertake that process. And so in those cases, we've helped develop new solutions to that. The image shown here is a tumor processing system. So to release the TILs, currently you have to carefully slice tumours manually, digest those tumours, and release the TIL cells. That needs to be automated if we're to scale, if we're to get control of that process, if we're going to remove variability of that process, and ultimately, in many cases, we're going to reduce cost in that process. And it's a slightly unusual problem because it spans two quite different worlds. The tumour is collected in the OR by a surgeon, given to a nurse in that environment, loaded into a consumable. That consumable is then sent to a much more familiar environment to many of us in the cell therapy space, a GMP environment, where it's loaded onto a system that needs to process that by a GMP operative. And so that's spanning different worlds, it's spanning different regulatory frameworks, it's spanning different expectations, and that's before we've got into the technological challenges that exist under that and achieving the biological efficacy that has to be delivered. So a fascinating project. The second is biomagnetic solutions, so that's part of the Gamma group. Many of you might know Phil Vanek. They have a very exciting ferrofluid technology for magnetic cell separation. And it requires an instrument to automate that process. Again, if we're going to achieve scale, if we get that into ultimately GMP environments. And so we've been developing for them a GMP system that will automate that process of magnetic cell separation with their unique ferrofluid technology. The third one's a slightly different business model. So this is one that we've developed as a TTP spin-out, which we do from time to time, and was born out of us asking a question of how many cells does a therapeutic cell sorter need to sort? Something of a tongue twister, but a question that we asked about 5 or 6 years ago. And the answer was more than any existing cell sorter could sort. So we began the journey of trying to develop a new technology to address that problem. It needed a fundamental new sort capability. So unlike existing cell sorters, it uses something called vortex-based cell sorting. Initially, we tried to do sorting by creating a microbubble and offsetting a cell in a microfluidic channel using that. Unfortunately, the forces that provided were not sufficient to achieve what we needed. And so we moved from that to using a bubble to shed a vortex, which then subsequently offsets the cell. And that is now going into beta trials with customers and has the capability in its final form to sort around about 2 billion cells an hour. The fourth one is Aglaris. They are addressing the challenge of how do we keep cells in the most effective condition to enable expansion? How do we keep them in exponential phase to accelerate their growth? And they have an innovative technology that utilizes some closed-loop control to keep the cells in that condition by looking at how that process is developing, and so they can get much better expansion through that system. That's the consumable for that system, and we're helping them take that product through to commercialization. And the final one is a company called Cell Fiber. So a Japanese company who are using a alginate-based extruded fiber, growing cells in the core of that fiber to protect them from the external bioreactor environment, whether that's high shear, as we're trying to undertake a media exchange, for example. And that allows for much more successful growth, much more protection of those cells, even if we're undertaking quite aggressive fluid exchanges. And that's a system there that we're now pushing ahead with in prototype form, will ultimately take to a GMP product. And behind all of that, we're also working on fundamental technologies to help progress the industry. So right back to preclinical models. So we are working on a novel micro-perfusion bioreactor at about 20 microliter scale that allows for things like model development, toxicology experiments, and optimization of the growth conditions before we move up to the more familiar scales. Analytics comes up a lot at events like this and remains one of the big gaps in moving forwards in this space. We're working a lot in particularly optical techniques, things like mid-IR and Raman, to get more information in real time out of bioreactors, as well as looking at automated sampling techniques. We have plenty of manual sampling techniques and manual processes, bringing together automated sampling and point-of-care diagnostic technologies to actually undertake at-line analytics to allow us to understand more about what is going on with our cell lines. And new enabling technologies, things like transfection, tools around electroporation and how we apply things like we've learned on cellular highways to be able to do much more precise cell-by-cell electroporation so that we can undertake more controlled delivery of cargoes into cells. And finally, future automation. We know that ultimately to get these products to patients, we need to go beyond the automation we have today. And we're very proud of the work that we've been able to do with other companies, are looking at the steps that need to happen, the unit operations that need to happen. But still, we need to bring these together to create the factories of the future, things like Amgen needed to create for the EPO line. And that is a big focus for us as we move forwards. I just want to briefly comment on what it takes to get beyond those prototype systems and put them into production. We've invested behind the capability to create pilot production facilities and be able to produce hundreds of instruments and thousands or even tens of thousands of consumables to get us to those first-in-human trials, sometimes those early customer studies, so that while we're building that confidence, that verification, that validation of those technologies in use, it's at that point in parallel we can be looking at transferring those across to manufacture, whether that's in a low-cost geography like the Far East or some other high-volume manufacturing capability, rather than pausing developments just at the point where we've shown promising early data to move to a transfer to manufacture. Being able to provide that early prototyping, those early systems to market, is increasingly critical in expediting these developments. So just return to where I started. In the last 8 years, I've been very pleased to see the progress that we've been able to make around the things that support the biology in this space. I guess one of the challenges has been the biology, as we've just seen, has moved ahead at a startling rate as well and continues to do so. So there's no sitting back. We have to continue to make sure we progress everything around this space that's going to bring that biology to patients. I've really enjoyed the journey that we've been on during that time, and I'm very excited about the future, and I very much hope that maybe some people in this room today will become people that we'll get to work with on that journey as we move forwards. Thank you.